Okay, so here I'm working as a senior scientist docent at the Aalto University. As you can see, the background picture of Aalto University. This is just one part of the university. It's like the arts and business department with some market area. So it's big university and one of the renowned university in world. It's ranked 130 on the top in the globally. So just briefly, I'm going to explain what our university is doing and what I am working and what my department is offering as a collaboration. So I'm mainly working on nano techniques. This is a one example of TM cryo TEM. Like if you can see in the screen at the left side, this is Vitrobot where we can prepare all the cryo samples, liquid or whatever samples you have. Once you prepared here, you transfer your samples to the cryo setup, and from here you can directly take in the TEM. So these facilities we have in our department, and these are one of the very popular facilities, one of the advanced techniques in our department. And then some other equipments like we have, this is normal TEM, which we work on environmental condition, not on the cryo. And right hand side you can see SEM. This is also one of the best and very advanced SEM we have. Not only one, just we have 5-6 SEM, and all the SEM have the different features. And for spectroscopy chemical information, we always do some Raman and other SACs and so many other techniques in our department. So this is just very brief introduction about department where I am working. And also this is the process how we prepare our samples for the TEM or SEM analysis. And the picture what you are seeing here, this is like a ultramicrotome. Here just we cut the samples, make the sections something like 100 nanometer or less or more. So, and we put on the grid and we take the sections and see in the TEM machine. Okay, so after this, now let's come on the topic for which I am here to discuss on that. So basically, you all know this radiation is one of the major issues, not only in India, but this is global issue nowadays because most of the global countries like US, UK, or European countries, they have their guidelines how they can follow the radiation, how much radiation they can consume in their everyday life. But India, at least, we don't have that guidelines. But I know ICMR is working on that. I was in JNU, so from that time, still ICMR is working with JNU scientists, and they are working on these guidelines. And hopefully, our India will get some well-known guidelines how we can use these cell phones and electronic appliances. So in picture, you can see this is some of my publications with US researchers, professors. The first one is from Professor Agarwal. He is one of the renowned personality in US. So we worked together in several papers, and here you can see there are different appliances, electronic setup, as you can see in the picture, like cell phone tower, wireless, laptop, TV broadcast, microwave oven, what we are using at our home. So they all emit some kind of radiations. And due to that radiations, for example, if you are using your cell phone near your head or keeping cell phone while talking in your trouser pocket, so it always affect your brain as well as your reproductive system, as you can see in picture. So generally, if you have long-term exposure with the testicle organs, so it may decrease the sperm count, decrease the sperm motility, sperm morphology, and also antioxidant activities. So this is not only the reproduction of brain, but this also affect throughout your body system, which you can see in the second image. And then we have something like a interesting pathway we created to represent how these radiations basically working. So once animal get exposure, after that what they are receiving some kind of signal, radiation signals, and it affect the membrane, and through membrane this induce the reactive oxygen species through oxidative stress, as you can see in the picture. So through oxidative stress, it is start causing several parameters related to your body parts, something like for sperm or brain, whatever you can see from the picture. This is mostly on the reproductive part. So it's affecting it damages the DNA. Once DNA damage, its cells become death, and this whole process, it leads to the cancer progression. So this whole process, if somebody is getting long term exposure, if I am right, so someone from Germany, they researched, like if someone is using more than 2 hours of cell phone, continuous cell phone uh, talk, so they have some kind of the brain temperature increase something like 2 degree. So it's something, it's very dangerous. So it's always better to restrict the call, take the short calls, and use earphones and all those things. And if you can see the another study here, they have shown different parameters. For example, viscosity, sperm count, motility, viability, and several others. So what we are seeing in this picture, from parameter 4 to 8, there is a significant effect on the sperm parameters. The person who is using about 4 hours or more time while talking. So you can see from the picture, who is not using, they have less effect. Who is using more than 2 hours or 2 hours, they have more effects. With increasing time, the effect is getting more. So this is the concept of this picture what we are showing here. Then when you go to buy some cell phone from the market, you always, it is a recommendation that you always check the SAR value. SAR is like specific absorption rate. This is like, specific absorption rate is like what uh, amount of radiation it is absorbing in your skin or in your body. So that gives the specific absorption rate. So it's always, you can see on the back side of the mobile phone box, it's always mentioned there what is the SAR value of the phone. So always try to take as low as SAR value of all the whatever cell phones you are buying. So always try to restrict this SAR value. And the limit is 1.6 watt per kg. That is the minimum limit what recommended. It should not more than that. And this is like JNU study what I did there. These are the measuring equipments, and here you can see in the flow cytometry study, there is a two parameters. One is sperm count and apoptosis. So in control, this apoptosis is less, but in exposed, you can see. Sorry, in control, this below side, this control is less apoptosis, but in exposed one, apoptosis is increased. And sperm count, this second one, is sperm count is very normal in control one, but in treated one, it's it's decreased sperm count. Then I can go through some more details about these radiation parameters. As you can see, this is cell cycle analysis, and this is very interesting study we published. It's quite old studies when I was doing my PhD, so I did all these things from there. So, like here you can see cell cycle analysis from flow cytometer. So this cell cycle basically it's representing the this DNA synthesis, how it's affecting the synthesis part of the DNA, because it's always this cell cycle is always depending on this synthesis. So if once its affect, as you can see from the picture, this S phase, which has significant difference if I can compare with the control and exposed one. So it's affecting the DNA synthesis phase of the cell cycle. So it shows like if there is some decline in the sperm count or some other parameters, so it's supporting that study. And then we did some TM, transmission electron microscopy study. So we took some direct sperm, animal sperm, with star rat sperm, and we take the images in TM. So this one is the control, A is the control, and B is the exposed one. So you can see the clear difference between control and exposed one. So A and C is the control, and B and D is the exposed samples with cell phone exposure. So it's totally distorted the head of the sperm. This whole area is the sperm head. which has been distorted by the radiation. And also this micronuclei sheath, they have like 9 frolar structure on that, which is completely distorted after exposure, after 2 hours of exposures. So these are very interesting studies which we obtained during that time. And then we, with the help of CSIR, I received some grant there when I was working in one of the university in India. So I received some grant, and from there I built this setup. And this is very, very novel setup. The concept of this setup is like, whenever we use our cell phone, we never keep at the constant position. So we always, take our cell phone at different angle. So with that angle, keeping that in our mind, that concept, we build the setup where we can monitor everything through computer. So this is computer controlled setup. So we set the temperature from, for example, 0 to 45 degree, and for 2 hours continuously this cell phone from in the box there is an animal, and from there cell phone is going through, like, 0 to 45 degree continuously, up and down for 2 hours. So this is such kind of exposures. It's showing how this radiation is affecting the physiology of the cells. So you can see again here, this is like I used 3G. At that time 3G came. So this whole study was on the 3G. Nowadays people are working on 5G because we have already in market the 5G networking. So from here we confirm that there is a DNA fragmentation. You can see in exposed if you can compare with the control and other groups. And here is the cell count and apoptosis, what it is showing here. And also this is like cell apoptosis. So in exposed one, you can see the apoptosis is very high. If I can compare with the case control or control. Case control is like animal is not receiving anything. They were just in the case. And sham exposed is like they were always going in the chamber, but without any exposure. And these are some genetical studies which expression of the genes, how is it's affecting the expression of the genes. As you can see in the exposed one and the control one, it's clear visible. Then we have published, this is also a part of the same paper, and here we checked some micronuclei, which is, very, very important parameters. Micronuclei is something like representing the chromosomal abbreviation. Any damage, any effect in the chromosomes, it's representing here. So you can see, like in case control, there is no damage. In sham exposed, you can see there is nothing there. But in exposed, we stain the cells with acrylyl orange, and you can see this orange color. It's the stain in the orange color is stain in the cells. It's showing clearly, and that we have plotted also how the effect is there. And there is one more interesting study. As you can see, right hand side, this is comet assay, and comet assay is very, very popular. At least that time when I was working there in GNU, so that time it was very popular. And comet assay, the concept of comet assay is like, if there is any damage in the cells. For example, if I am taking some brain cells, if there is any damage in the cells, so there is always some kind of breakage in the membrane. And the DNA, what is the concept of comet? If there is any damage, so DNA will come out from the cells, and that DNA, what we do, we take these cells and run in the electrophoresis, and electrophoresis, this DNA start migrating from the cells. So the shape comet, what you are seeing here, you can see like the shape, that is the DNA, the migration of the DNA here from the cells to the other side. So this is confirming that there is a DNA damage, and you can also analyze here apoptosis, several other things. I mean, DNA single strand break, DNA double strand break, whatever you want, you can just check from here. And also you can make the analysis, how much significant differences are there in the damage, what you can see. So this is, first one is control, sham exposed, and again exposed. So there is no difference, no damage in the cells in the case control or sham exposed, but in exposed you can see the comet shape of the cells, which representing the effects. Then this is also very interesting study we obtained in the GNU only, and we did several enzymes to check how these radiations are affecting, because these enzymes, especially protein kinase C, PKC, which is highly responsible for the cell signaling uh, factors. So if there is any changes in PKC or creatine kinase, these are like cell signaling, which representing the cell signaling pathways. So if there is any changes in these enzymes, it's totally changed the metabolism of the body or cells. So if you can see from the picture, like there is a significant differences in these both parameters. And also the third one, melatonin, as we all know, this melatonin, basically this is where this brain produces in response to the darkness, or this circadian rhythm, this night and dark system it maintains. So this parameter is very important to check how these radiations are affecting our uh, sleeping things, what we are, it's like daily basis things. And in continuation to that, this is also the part of the same paper. You can see like there is some antioxidant enzymes. This antioxidant, why we use antioxidant. Antioxidant is very important for our body, because whatever damages you are getting from outside sources, for environmental or from anywhere, so these antioxidants work as a protectant. It always protects the free radicals formation in your body. It always protect from any damages which may occur in your body, and they may produce some kind of diseases. So these antioxidants are very important. If there is any changes in antioxidants, it shows like your body's cells are affecting highly with these radiations. As you can see, like there are significant differences in all, and especially this ROS parameter, reactive oxygen species, which is something like free radical formation. If there is any oxidative stress, for example, if I can give you one simple example, suppose this is my hand. If I can hit it here, so it will somehow it will start breaking the there is some single strand DNA break, or some kind of damages it will start, because I am giving a stress here. So due to that stress, but these are not stable stress, of course. These are like it will repair very fast. So ROS is basically what it's giving. It's giving the analytical method. It's basically it's the parameter from where you can decide there is effects or not. So this basically measures the free radical formation in your body. If there is any kind of stress, it's your body is getting from outside source. So this is very important parameter. Then there is another setup. Just I talked to you about the cell phone, but now this microwave oven which we are using in our home, but they are safe. Those microwaves are safe. I'm not saying that those are not safe. We use this microwave just for the exposure. It's not like we are using this microwave and there is some kind of emission. I'm not saying that. We modified this microwave to expose the animals. You can see it's connected to the chamber, and their frequency is 2.45 gigahertz. So animals, we always kept animal in these boxes. You can see these boxes. So there are 6 animals at a time. These are very big exposure setup we had there. So we always expose animals at a time, 6 animals at a time. After 2 hours of exposures for certain time period, we sacrifice the animals and we go through the several parameters to check how these radiations are affecting that. So here also you can see the clear changes in the DNA parameters, like this comet is, the shape of comet is very clear here. So it says like there is a DNA damage in the cells. If I can compare with the in the control group. Then this study is also one of my PhD student. I was the co-supervisor actually. So she did really nice work, and she published 3-4 papers, and that one paper is a part of that. So she was working in Rajasthan University with Professor Shishodia ma'am, and she developed some technique to measure the memory loss in the animals. Uh, this is also very important study, because with the aging, mostly with the aging, people are losing their memory power. So in animals, to check how these radiations are actually affecting the memory, that is very important. So if I can just play, I just took this video from the internet, so you will just come to know how this work in short. So this is first time training animals taking more time to find the platform in the water. So once animal trained, so it took less time. This is for the normal animal. I'm talking about the normal animal. And the third time when we proceed, so the animal reached very fast to the platform. Uh, this is for the normal animal, but for the exposed animals, they took always more time, as you can see from the figure. Like this is the exposed animal, and this is the normal animal. So this, let's complete this video. So normal animal, it always take less time to reach the platform, and exposed animal, microwave exposed animals, it always take more time to reach the platform. So with the increasing days, at some point it become normal, but it's somehow affecting the this memory also, this microwave radiation. It's very high power, actually 10 gigahertz. It comes under very high power radiation. And these are the histological measurements of the uh, animal tissues, and where we check all those parameters which are very very important to check how this memory loss is happening. As you can see from picture, especially, like there is a darkness in the cells here, and there is these are like control cells, and these are like treated cells. So it's very clear differences you can see from the both side of the pictures. And this is also a very nice study. This is a output from my CSIR project, which I obtained, which I hired some researcher there, and they obtained the study. This Ramatar Meena, and he is now actually assistant professor. He's working as assistant professor in GNU now. So he conducted that study there, and he obtained very nice data on male reproduction, where he found like there is a significant damages in the microwave exposed group. But the good thing about this study is like he also used some kind of antioxidant, and melatonin uh, he considered as an antioxidant to protect the radiations, to protect animal from the radiation. So melatonin, it's what doing, it's decreasing, as you can see from picture. In these, most of the cells are damaged, but in melatonin microwave, most of the cells are more normal cells. If I can compare with two others pictures on the top. So this is a very interesting study which we obtained there in the. And now I'm showing my studies which I did in Finland. This is these studies are from my first postdoctoral study. I worked there as in University of Eastern Finland, and there I generated this is very very interesting paper, and it took me so long time to generate the data, because there is a long time incubation. As you can see, 45 days of incubation. So this study is showing like I exposed the cells. This is a very low frequency. The frequency which if I can explain you in very normal way. So once when you go in some mall for the shopping, you always see those magnetic plates which are standing on the door of the mall. So those plates or wires which we have in our buildings, they are generating 50 hertz of magnetic field. So these magnetic fields, how they are affecting our daily life. So this whole study is based on that. So what it is showing, basically, we exposed cells, neuroblastoma cells, once, something like, if I remember right, so I exposed them for 2 hours or something like that, and after that, then we put them for the incubation for 15 days, 30 days, and 45 days. Same cells. We have not treated again. So the concept of this study was to see how these radiations are affecting in the progenies. So you can see, like after 45 days, also these cells were containing some kind of effects, radiation effects. So this concept is known as genomic instability, which is going from parents to their progenies. So this is very interesting study. I'm not going very detail in this, as like this is very big study, so it will take so much time to explain all. But here also we used some kind of antioxidant, as you can see, NEC, N-acetylcysteine. This is antioxidant which not worked for against the radiation, so it did not block the effect of the radiation. So this is another study we published in Royal Society Journal. Just so this study is basically I did in UEF only, University of Eastern Finland, and here you can see like the data what I'm showing. These are like from the micronuclei, which I already explained you. Micronuclei is representing the chromosomal aggregation in the cells. They are affecting the chromosomal level. So with the increasing, and these 0, 1, 5, 10, 20, these are the concentration of menadine. We also used for this study the menadine. Menadine is like, it's free radical generator compound. It generates the free radicals. It induces the oxidative stress in the cells. The concept behind this to use menadine was like 50 hertz magnetic field itself not very high to affect the cells. So that's why we used menadine to see how this menadine is affecting with the radiation. So what is happening here? You can see these are the control. These are like only without menadine. So these are the but with magnetic field also it's showing some kind of effects, significant effects in the cells. And this is the mitochondrial superoxide production, where we have also seen very significant differences in all the concentration of the menadine. And just I took this slide as a part of radiation. So nowadays most of the people they are talking more about this COVID things. So when someone has published this paper like this, in COVID, this radiation is playing very important role. I just come to know yesterday. I was not knowing that this radiation is also enhancing the effect of COVID, and it's like the effect is almost double once when it combined with the COVID infection virus. So from this paper, it's very interesting paper to see how this in this pandemic. So I can say like the who has some severity problems, they might be also facing some radiation issues with that. Maybe this radiation is helping to cause the severe condition for the patient, COVID patient. Then this is also our very recent study we obtained. And this is on the radiotherapy. There are several treatment methods, cancer treatment methods, radiotherapy, chemotherapy, so many methods. So people who are thinking like they are fine with the radiotherapy, they are not right, because after this radiotherapy always have several side effects. So this study actually showing last 20 years of studies how this radiotherapy has the side effects after I mean at one way you treated well with cancer, but at another side you have some side effects also. So this whole study what we obtained here to check how in last 20 years of study how radiotherapy is also affecting the human being. Then this is my very recent work which I am doing in Aalto University. This is on the antioxidants what I am using actually. I'm in fact I'm isolating all the antioxidants from the plant species. Here we used willow. This piscine is very new compound which we used in our study, and it's very simple method we applied here. As you can see from here, this is from one paper from our group. we extracted some compound from the willow bark through hot water extract. It's very simple method. Just we put all the bark in the hot water 80 degree, and we take those extracts and do several chemistry for that. Once we come to know what compounds are there exact in that, after that I applied the bioinformatics to check how they are biocompatible with what diseases. So once when I come to know this Piscine is very effective for the Alzheimer patients, then I started working with neuroblastoma cells, and after that we found with this compound Piscine, like it's showing very promising effect, very as a neuroprotectant. So you can see like this is the control, but we used Menadine. So this compound is working very nicely against the Menadine. It's decreasing the effect of Menadine, this Piscine. So this is just Menadine, this black bar, and this is Piscine plus Menadine. So it's decreasing the effect of Menadine. This blocking the free radical formation in the cells, and this is something like very important study we published recently in India also. And then this study is from my PhD student. She has just published paper, and she did really nice work. She used vitamin K2 to treat the neuroblastoma cells against streptozotocin and melanoidin. These both compounds are free radical forming compound, so they induce the free radical in the cells, and that's why we used vitamin K2 to check how it's a is there any blocking effect or not against these two compounds. And we found like from picture you can see it's very clear in control group there is nothing, no any stain. CHOP is like endoplasmic reticulum marker, stress marker. So if they stand with the cells, it will show like it always shows there is some effect, there is some damage. If it's not stained, so it's like control, there is no damage. So in control there is no stain. In vitamin treated cells there is no stain, but in this compound you can see there is a clear stain. Uh, then we used this vitamin with this compound, so there is again no stain because it's blocked the effect of this streptozotocin. And again alone menadine it's causing the effect. You can see there is a big amount of CHOP stain, and again with menadine with vitamin it's again neutralizing the effect of that. So this is very interesting study, and we are still continuing with this study further, and we are also working on more advanced vitamins which are now in use actually. And then this is also a part of the same paper where we conducted several parameters to see, especially for the Alzheimer disease related parameters. You can see this is total tau protein which is affecting with different combinations with alone with streptozotocin and with vitamin. Then I mean you can check all those how significant differences we can see here in all the groups. So this vitamin K2 it's really protecting the cells with antioxidants. And this is something different from the other part, but this is I am showing because here most of the students are from the biotechnology background. So this is a part of the technique which I am using here. This is one of the advanced technique which I generated here in Finland. It took me something like 1 year to generate this data. So this is from the wood cell. What you are seeing here, this is from the wood cell corner. This is wood cell corner. So what I did, I took the 3D images from the cryo TEM. I make some modeling, and I took something like 40 images, and then I combined all the images in one image. So it has something like some nanometer thickness. So after combining them, you can see the cells how they are looking. Two layer of the cells, where this like sorry. So This is what you can see, like here, these are the, with microfibrils, and these are something which is showing very important study here what we conducted in our department. This is also a part of different plant species, because I'm also working on the plant species. So this is something very different from the other things. So this took me something like 1 year to generate all this data. This is the cell cell wall. So this is just showing how this, there's different cell wall layers in the cells, in the wood cells, and they have the different orientations. So just we check here how these orientations are there, and these are they have very important applications actually. And this is also another study, so I'm not going in very brief of these things. This is from the one of the technique what I showed you in the presentation, like the Raman technique, which we presented, which we generated here for the wood sample. So these are the wood cells what you are seeing here. So this is also very important study which we got that. So these are some of my recent publications which I published in last 1 or 2 years. Mostly they are very new publications. So just very selected one which I recently we got published in molecular cancer where impact is 27, and then several other papers as you can see from the list. Then overall, I have something like 110 papers now or more. I have edited several books. These are some of the examples of those books, and several other books are coming soon. Okay, so this is all about my presentation.
Kitosera sir, Dr. Kesri, for giving such a simple and wonderful method to the student now how to tackle the influence of radiation on their psychology and making their life better. I am confident that this will definitely help the student forever. Now, dear student and faculty members, अगर आपकी कोई query हो तो just raise your hand in chat box and technical team will connect you.
Hello, good afternoon, sir.
Yes, good afternoon.
Hello, good afternoon, sir.
Yes, good afternoon.
Am I audible, sir?
Yes. Go ahead.
My question is just like every coin has two faces, like every process has some benefits, some disbenefits, or some harmful effects. We have always been studying and hearing that radiations has some side effects on psychology or on the personality of an individual. Do we have any benefit of radiations?
Yes, we always have benefit. We are connected throughout the world. That is the. I'm not against the technology. I'm not saying I'm also using the mobile phone and everything.
I mean, ma'am, sir, my question is that do radiations have any positive effect on the personality of the individual or the body or the psychology?
Yes, I mean with the exactly. I exactly I'm not able to answer that because I never worked on that part. But of course, there is always some positive and negative things, pro and cons of everything. So if you can explore that, so of course there is a positiveness of this radiation also, because as a psychology, if I can say, like people are connected with cell phones and several other equipments.
Yes.
Today's, if I can talk about the technology, it's totally digital. So if you do, for example, just see if you don't have these setups, so how you will feel. your
Okay, so
psychology will
it
completely
can be in
change with
this.
that.
As of now, we can say that it can be in the sense of mental satisfaction.
Yes, something like it's mental satisfaction. Of course, whenever you feel loneliness, you always take yourself phone and start searching something on that. So this always give you some mental satisfaction.
Yes, sir.
Satisfaction.
Yes. Thank you. Thank you.
और किसी की कोई क्वेरी हो तो रेज योर हैंड. Now coming to the end, I request Dr. Nabil Ahmed, Associate Professor.
Ma'am, Shivani
Yes.
Joshi has raised her hand.
Okay, okay, ठीक.
We are making her the co-host.
Okay.
Shivani,
Yes,
you can
ma'am.
ask your query.
Okay, ma'am. Hello, sir. I have a question.
Yes.
Does medical diagnostics also affect our body?
Yes, there are so many equipments. I have already mentioned, like this radiotherapy or NMR or MRI or X-ray. All these equipments, they are releasing high amount of radiation. So that's why there is a guidelines or recommendation from the doctors that if like for example this X-ray, you don't they never allow you for one or two times, more than two times to take the X-ray. So these radiations are, in these medical equipments, they are releasing lots of radiation. And if you can search on internet, so you will find so many literature on these issues also, on which I think many people are working on this medical appliances radiations.
Okay, sir.
कृति, अब किसी की क्वेरी तो नहीं है? कृति मैम, अब तो क्वेरी नहीं किसी की. Hello?
No, ma'am. I think there is no query anymore.
Okay. Now coming to the end, I request Dr. Nabil Ahmed, Associate.
Sophie sir had a query.
Okay, okay, okay. Chal.
Hello.
Yes, hello.
KC sir, very good afternoon.
Good afternoon, sir.
Sir, myself, Dr. S.K. Srivastava, speaking from IFT University, Department of Biotechnology. Sir, first of all, I will be very thankful for you for providing the very true information like the research value of the effect of the radiation on the living system. Sir, my question is there any time parameter of exposure, like short time exposure, long time exposure?
Yes, actually, I already mentioned in presentation, like there is some German study. They have conducted some research and they said, like, if you are using your cell phone directly without using earphone or some just direct cell phone for more than 20 minutes, so your brain temperature will increase. I mean, in logical, you also know that if you are using your cell phone for 30 minutes continuously, so some heating things start there.
Yes.
So it's something like, so time duration is restricted, so it's always good if you are using less than 20 or less than 15 minutes.
Yeah, in your study you have mentioned like exposure time, something like that. You have a study on the rat.
Yes, we have conducted several studies where we have done several things with lower dose, low time, something like 1 day to 30 days.
Yes.
And with increasing time, the effects, the effect size is getting more. So we conducted those studies in our lab.
Okay, sir. So my one question is that can we reduce, like we can use any technology that can reduce the radiation from the electronic sources?
There are so many cheap setups, but I am also actually associated with one of the international committee as a commissioner.
Yeah.
In that committee, there are so many renowned scientists are there, so they always recommend not to use these chips. Always in most of our meeting, we always say like these chips are not safe. They are just they are in market just to earn the money. From there, at least there in literature, you will never find anything that those are beneficial or inhibiting the effect of radiation or blocking the effect of radiation from cell phone, emission of radiation from cell phone. So
yeah. Is there not any technology like any chip or any material like which can absorb the radiation?
there are so many things. People are like there are many nanomaterials based on the fabrics. People are using fabrics, seat in this mobile phone. Those covers. They always recommend to use those mobile phone covers bags, and in those bags, there are some kind of fabrics they use coat inside the purse, and then if you can put those cell phone inside that cover, so this radiation will not come out, so it will not interact with your body. But these are like, just I'm telling you, these are not in literature, these are not, there is no scientific evidences on these are just on the internet. So I never recommend to use those appliances, at least if you do not see any scientific evidences on that.
Yeah.
It's better way to, we always recommend, our recommendation is like just restrict your call, and if you are taking longer calls, so use headphone.
Okay.
Earphone. Keep your cell phone as far as possible.
Okay. So thank you, sir. Thank you so much.
Most welcome.
Now coming to the end, I request Dr. Nabil Ahmed, Associate Professor and Head School of Biotechnology, to propose vote of thanks. Over to Nabil sir.
He is muted, I think.
I'm audible?
Yes, now.
Okay. There is. Yeah. Thank you, Dr. Rashi Srivastava, madam. Well, we all know that radiation are harmful for health, yet the time was spending chatting and browsing through our phone is ever exceeding. I think both physiological stress and ionizing radiation can cause very detrimental effects on humans. With these words, I would like to extend a very warm good evening to all of you. Honorable Chancellor Sir, Shri Rajiv Kothiwal, Pro-Chancellor Sir, Shri Abhinav Kothiwal, Vice-Chancellor Sir, Professor M.P. Pandey, Pro-Vice-Chancellor Sir, Professor Rahul Mishra, Registrar Sir, Professor Sanjeev Agrawal, Directors of different school, faculty members and dear students. On the behalf of School of Biotechnology, IFM University, I, Dr. Nabil Ahmed, extends a heartfelt thanks to the keynote speaker, Dr. Kavinder Kumar Kesari, Senior Scientist, Department of Bioproduct and Biosystem at Aalto University, Finland. Thank you, sir, for delivering an outstanding content on influence of radiation on human physiology. I believe this lecture was beneficial for all of us. I would like to extend my sincere thanks to all the authorities for their kind support and their encouragement during the organization of this program. I would also like to extend my sincere thanks to the faculty members of the School of Biotechnology, especially Ms. Ritika Saxena and Ms. Kriti Shinet. I must thank everyone present here for their interest, support, and patience. Thank you. Thank you so much.
Thank you, sir. Once again, thanks to all present here. Now we shall end the webinar with the words that if we conquer the psychological issue, we can conquer the whole world. At the end, please fill the feedback form which is in the chat box. Thank you, sir.
Thank you so much, ma'am. Thanks a lot.
तो
Thanks
जरा
for
सर
everyone.
कुछ फिनिश मुझे भी आ गई है सर लैंग्वेज मैंने भी सीख ली
मेरे
है
से ज्यादा आप सीख लिए हैं। मुझे 9 साल हो गया लेकिन अभी तक आया नहीं, लग रहा है आपने ज्यादा सीख लिया।
यस सर आपको जो बुलाना था सर
जी 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 जी।
थैंक यू सर
So much. Thanks a lot, ma'am.
ऑल में लीव द वेबिनार
Okay.